everyone, I'm John Odipio. I work with Naro and I'm based at Namlonge. I'm excited to tell you a story about how I traversed 3 continents to demonstrate efficient gene editing in cassava. Cassava is the single most important commodity which gives the highest return on investment. I grew up in Vora sub-county, the food basket of Arua. In Sunguru village, where my dad Adiko Isaiah grew cassava, everyone in the community relied on that most important commodity. It was from cassava that our food came, and at the same time, money that paid our fees while I was in Vora Secondary School and Vara was generated. So when I went to the university on government sponsorship, I still got my out-of-pocket allowance from cassava. So after finishing my master's degree, I started working with cassava program at Namlonge, and I soon learned that while farmers need stem cuttings of a good variety to start a new crop, breeders need cassava flowers in order to generate new cassava varieties. And I also learned that cassava does not flower at the same time. So because of differing time of flowering, scarcity of flowering makes breeders to take up to 10 years to develop new cassava variety. I promised myself that I will do something to generate information to understand why this happens in cassava. So in 2014, I got an opportunity and flew from Uganda in East Africa via Ghent University, Belgium in Europe to St. Louis, Missouri, USA in North America. This flight was very important because it marked the beginning of a 5-year PhD challenge, during which I had to find answers to many questions. One of the questions was whether gene editing, which is a new technique for modifying genome of organisms, can be used to speed up research and development of new cassava varieties. Gene editing uses CRISPR derived from bacteria to cut the DNA, and once the DNA is cut, the cell has the ability to stitch together the cut DNA so that new genetic information is constituted in forms of addition, substitution, and deletions. And these genetic changes take place in a particular and exact genomic location, and they are similar to what you will achieve when you do conventional breeding and mutation breeding. And therefore, the products of genome editing don't have to be regulated. So having grown in a farming household, I was aware that it takes long to receive new cassava varieties, exactly more than 10 years. And also, at the research station, breeders were having problems to develop new cassava varieties because cassava would fail to flower at the same time because they have to mate different parents to produce new cassava varieties. So with gene editing in place, I wanted to identify what genes within cassava were making it not to flower at the same time so that breeders can do their development of new varieties faster. I was very glad to receive full sponsorship from Donald Danforth Plant Science Center. While I had the full sponsorship, I had challenges to deal with. First of all, the climate, the change in climate from tropical to Mediterranean was hard for me because the temperatures during the summer were so extreme. Then during winter, it will get so cold that I had to always have heavy jackets, which made me very uncomfortable. But I had to live with that. And in addition, I left my family in Uganda, meaning that my wife, Davbezu Mole, had to take care of our 2 sons and will only keep in touch through phone calls. And the last and most important challenge was the complexity of the science that I had to deal with. So having waited for 4 solid years for an opportunity like that, I was determined to ensure that everything worked so that I will come home with the good news. So I started the work under the supervision of Dr. Nigel Taylor, who, under his good leadership, constituted a good team at Donald Danforth Plant Science Center, and at the same time, my university supervisors, Professor Gayet and Mark Hedge, together with Dr. Titus Alsai of Naro, these people worked together to ensure that I had comfortable environment to start with. So as I worked, I would sometimes fail, but I had people to ask and guide me through. And after 2 years, I finally got the answer I wanted. I was able to produce cassava that lacked the green color because I had successfully used gene editing to turn off the function of a gene called phytoindesaturase, which is responsible for the green color in cassava. This result meant a lot because I had proved that you could effectively do gene editing in cassava, just like is known to happen in rice and maize. So out of excitement, I ran to my supervisor's office. Reaching his office, I was so tired. I looked at him. He asked, "John, what's wrong?" I said nothing. He asked again, I said nothing. So the third time he asked, I said, "The gene editing has worked." When I said gene editing has worked, he said, he moved out of his chair and he was very excited. He said, "Congratulations." So I totally disorganized his program at that time, and I had to take him down to the tissue culture lab where I showed him the result. And while we were there, we kept discussing about the way forward and how we'll now use the experience gained to address flowering challenges. So from that point, I went on to use the same gene editing, the experience and the skills gained to look for genes within cassava which might be responsible for delayed flowering. So after 1 year, I was successful and I produced cassava of TME204 cultivar, which could flower within 2 months instead of 10 months under field environment. This meant that the same knowledge of gene editing could now be used to make other cassava varieties which have never flowered or will take long to flower, and therefore breeders have not used them in the breeding to flower at the same time as other cassava varieties, so that new varieties will be developed faster, to be precise, within about 5 years instead of waiting for up to 10 years. And what it meant was that the knowledge generated will help other crop commodities to be modified, and therefore we get super crop commodities which are resistant to diseases. Maybe they can be resistant to climate change, they might be more nutritious. And when farmers grow such commodities, we expect that in future they will get more yield, more income, and therefore improved livelihood within space of 5 to 6 years instead of 10 years that they will have previously waited for. And for this to be realized, African scientists have to be supported by their governments through investments in research so that they can innovate and generate solutions for the problems affecting African crops, animals, and also diseases affecting human beings. But this will also have positive effect if African governments adopt science-based regulation of products from technology, biotechnology, so that these products can eventually reach the farmers. And therefore, my journey across 3 continents to demonstrate efficient gene editing was worth the effort.